Welcome. You're at the cutting edge of cardiovascular sciences. I'm John Cooke. I'm professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist. And joining me in our studio today is Dr. Lee Lai, who will introduce our speaker.
Good morning. So today it's my honor and the privilege to introduce our esteemed speaker, Dr. Matthew Speight. Dr. Matthew Speight is an associate professor at Harvard Medical School and the lead investigator in the Center for Experimental Therapeutics and Reperfusion Injury Department of Anesthesiology, Perioperative, and Pain Medicine at Brigham and Women's Hospital. He got his PhD training in biophysics and physiology in University of Louisville School of Medicine and got his postdoc training in Brigham and Women's Hospital with Dr. Charles Sirhan. In 2009, he became assistant professor of medicine in University of Louisville School of Medicine and finally joined Harvard Medical School and Brigham and Women's Hospital since 2014. His research program is focused on elucidating molecular mechanisms underlying the transition between resolution of inflammation and the tissue repair and regeneration, and to understand how these mechanisms might be altered in chronic inflammatory disease, including cardiovascular and metabolic diseases. So Dr. Speight, welcome to our show.
Thank you very much for having me. I'm honored to be here and to share some of our work with you.
Well, thanks, Dr. Speight, for joining us today. I see you made it to your office this morning. I understand there was quite a snowfall in Boston last night.
Yeah, that's correct. Luckily, it was on Sunday instead of Monday morning. So, but by this morning, everything's back to normal completely.
Yeah. Well, it's great to see you, and looking forward to your talk. Inflammation and the resolution of inflammation plays such an important role, generally speaking, in regeneration and wound repair, and in particular in the heart. So we're very interested to hear what you have to say about inflammation and its resolution in the setting of ischemia. Tell us, Dr. Speight, before we launch into your talk, how did you come to work on this area? What stimulated you to work in inflammation and
Yeah, so during my graduate work, I was in the division of cardiology, actually. So we were interested in understanding the contribution of oxidation of lipids to inflammation that develops in the context of atherosclerosis, for example. And we began our work at that time studying non-enzymatic oxidation of lipids and how those are metabolized to less reactive products such as alcohols. And so that sort of like initiated my interest in lipids and how generation of new molecules from parent polyunsaturated fatty acids could modulate inflammatory processes and contribute to cardiovascular disease, either positively or negatively. So upon completing my graduate work in that area, understanding lipid metabolism, I became interested in understanding how enzymatic metabolism of lipids could lead to distinct lipid mediators that could be signaling molecules to activate innate and adaptive immune cells, and what their roles in modulating immunity and tissue repair and those processes sort of generally, as well as in the context of cardiovascular disease, might be. And I came across the work of Charles Sirhan, who at that time had discovered a new class of molecules involved in resolving inflammation, and I thought that was a really interesting area with a lot of therapeutic promise. So then for my postdoctoral studies, we initiated work to elucidate structures of new molecules that are involved in resolving inflammation and to determine whether those could be agonists to develop new classes of therapeutics to resolve chronic inflammation and enhance tissue repair.
That's great. I know that's it's a little bit off target for today's discussion, but what you just said reminded me of the history of oxidized lipids and their role in atherosclerosis, and from the great work done at in UC San Diego, for example, and the idea that oxidized lipids were driving atherosclerosis. I'm sure you've thought about this, although you're not discussing it today. How is it that antioxidant therapy has not achieved the promise that would have been foretold by the work with oxidized lipids and atherosclerosis?
Yeah, I mean, that's sort of where we entered when I was a graduate student, and sort of understanding why some of those therapies had failed. And, the whole antioxidant system is intimately linked, to sort of prevent perpetuation of radical propagation. And, I guess so being able to interfere with that specifically at specific sites and understanding how those products modulate immunity and things like that were sort of interest to us. So rather than taking the approach of just inhibiting oxidation, we were trying to study how those products that are generated are then metabolized, and whether we could modulate that metabolism as a way to reduce their toxic effects, given the failures of those antioxidant therapies. And I think the reasons for that are many, but that was our sort of main interest at that time, is can we find new ways to modulate these products that could not be successfully modulated with just general sort of antioxidant therapies. But there are approaches that have been shown to modulate those oxidation products that have gone into clinical development, from what I understand, such as antibodies and things like that.
Well, thank you. Well, I know you have a lot to tell us today, so maybe we should go ahead and get launched into your work.
Okay, sure. Yeah, let me.
Oh, while you're doing that, let me just remind our listeners that if you want to ask questions of Dr. Speight, go to pollev.com and enter DeBakey, and then you can respond to that activity. So if you're on your computer, go to pollev, that's POLLEV.com, and enter DeBakey. Or you can join us by text. In this case, you call 37607 and text DeBakey to that number, 37607, and text DeBakey, and then text in your message. That'll get to us, and we'll ask Dr. Speight your question. All right, Dr. Speight, the floor is yours.
So I guess I'll first check, are you able to see my slide?
We can.
Okay, great. So today I'm going to be talking about some of our work on inflammation resolving lipid mediators and their role in tissue repair and vascular biology. And just as a sort of brief outline, I'm going to give a very brief overview of the role of lipid mediators in inflammation and its resolution, because it's something that you probably don't hear about as much related to, in terms of regulating immunity. We hear a lot about cytokines and things like that, but lipid mediators are a little more underappreciated. Then I'm going to talk about some of our published work on the role of pro-resolving or inflammation-resolving lipid mediators and their roles in facilitating tissue repair, in part by regulating macrophage phenotype transitions. And then the last component of the talk, I'm just going to briefly touch on some recent unpublished data regarding the transcriptional regulation and downstream function of a pro-resolving receptor called GPR18 that we think could be a potential therapeutic target in the future. And then in just the last couple of slides, I'm going to highlight some new data showing that this receptor may have a role in regulating endothelial function in addition to its roles in regulating immunity. So as I noted, our lab is sort of broadly interested in understanding how inflammation resolves and how resolution interfaces with the next phase of tissue repair. So if you think back to the just general wound healing program that you see in your basic pathology textbook, you know that inflammation is one of the early responses to tissue injury. And then over time, this transitions into a reparative or proliferative response, which in this example in the skin would lead to a reepithelialization to reestablish the protective barrier, and also reestablishment of blood flow through angiogenesis. Thanks. And a provisional matrix that then goes on to be remodeled during the maturation phase for an extended period of time. And of course, the inflammation phase itself is also comprised of multiple distinct temporally regulated events. Most notably at the beginning, there's a edema that's associated with microvascular permeability, and this is associated with the subsequent infiltration of neutrophils, which are our first line of defense against an invading pathogen, for example. but these are relatively short-lived in the tissue, and they undergo apoptosis pretty quickly. And they are cleared by macrophages that are derived largely from monocytes that infiltrate into the tissue. And these macrophages take up apoptotic neutrophils and other dead cells in the injured tissue in a process called efferocytosis. And unlike neutrophils, macrophages persist in injured tissues for a much longer period of time, and they adopt a whole spectrum of different phenotypes from these sort of extremes, from a pro-inflammatory phenotype to more of a pro-resolving phenotype that starts to initiate these next phases of tissue repair. And I just want to point out, I have this term lipid mediator class switching here at the top to emphasize that there are distinct lipid mediators that are associated with each one of these distinct cellular responses. And I'll talk a little bit more about that as we move on. So you can see the normal acute inflammatory response that I just mentioned in this panel here. But I wanted to emphasize that in several chronic diseases, such as cardiometabolic diseases like type 2 diabetes, obesity, cardiovascular disease, several of these distinct cellular events can be altered and lead to persistent inflammation or defects in resolution, such as prolonged neutrophil survival, coupled with defective macrophage efferocytosis, so impairments in the ability of macrophages to clear these apoptotic cells. And this leads to a persistence of leukocytes in tissues. And this is most notable in a diabetic wound, for example, where you can see a persistence of neutrophils that, if not rapidly cleared, can start to actually cause injury to healthy tissue. And again, coupled with defective macrophage efferocytosis, these cells don't get cleared and they undergo post-apoptotic secondary necrosis. This leads to persistence of an open wound, so epithelial cells are not able to migrate in and close off the wound and protect against pathogens and water loss. And also defective tissue perfusion is also associated with diabetic wounds. You could also see very similar events occur at the level of an atherosclerotic plaque in an artery, for example, where it can sort of be viewed as a festering, non-resolving wound, where there's also a defect in clearance of apoptotic cells, a persistence of the leukocyte infiltrate that over time can alter the tissue repair response and lead to a thinning of a protective cap and a rupturing of that, leading to thrombosis and downstream complications. So this is why we're interested in pinpointing how each of these phases are controlled so that we can sort of intervene and rescue some of these so that they may be beneficial in cardiometabolic diseases. So as I noted, lipid mediators play key roles in both the initiation and resolution of inflammation. Most of you are probably familiar with the arachidonic acid-derived prostaglandins, for example. So arachidonic acid is a polyunsaturated fatty acid that is converted to a variety of downstream mediators by cyclooxygenase or lipoxygenase enzymes to generate mediators that regulate, vasodilation, vascular permeability, such as the cystenyl leukotrienes. Leukotriene B4 is a very potent neutrophil chemoattractant. So a lot of these mediators are involved in the initiation of inflammation and contribute to the cardinal signs of inflammation. In contrast, as this time course evolves, you can see that neutrophil infiltration eventually ceases, and this is due in part to the production of new classes of mediators that we call pro-resolving mediators, such as the lipoxins and resolvins, that, like the pro-inflammatory mediators, are also enzymatically generated from polyunsaturated fatty acids. Some of the precursors are omega-3 polyunsaturated fatty acids. And these mediators feed back to stop excessive neutrophils from coming into the tissue. And they also have distinct actions in promoting macrophages to clear apoptotic cells. So these are distinct agonist actions that ultimately give signs of resolution, which would be clearance of phagocytes from tissues and engagement of tissue reparative programs. So over the years, it's emerged that several chronic diseases are associated with imbalances in these mediators, such that there's elevated or persistent levels of leukotrienes, for example, and that when added back, the pro-resolving mediators can dampen that and then help to resolve established inflammation. And from a therapeutic perspective, you may know that blocking a lot of these enzymes with inhibitors is sort of the mainstay approach, such as the non-steroidal anti-inflammatory drugs. But this can, in some cases, have side effects, particularly for wound healing. And it's important to note that pro-resolving mediators, unlike a lot of the immunosuppressive strategies, have actually been found to enhance certain aspects of host defense, such as bacterial uptake and killing, while at the same time turning off cytokine production. So this may be sort of a new therapeutic approach that's not immunosuppressive and that does not impact or negatively impact tissue repair. So as a very straightforward demonstration that these pro-resolving mediators may sort of interface between resolution and tissue repair, we carried out a very simple model where we create an injury on the back of a mouse using a punch biopsy tool. And this wound can be collected, and we can assess production of lipid mediators by mass spectrometry. And shown here is a metabolome, if you will, of one particular polyunsaturated fatty acid called DHA, which is converted into a variety of downstream products, such as the D-series resolvins. And what you can appreciate is that there was an increased production of some of these pathways immediately upon injury. This is day 1 post-wounding, and that some are more prominent than others. And then if we just asked a very simple question, well, if they're generated during an injury compared to non-injured skin, if we add them back, what do they do to the tissue repair response? And you can see that if we just add them back topically at the initiation of the wounding at day 1, you can see, shown here for an example with RVD2, that we're able to enhance wound closure, decreasing the time to 50% closure by about a day, which is substantial when you're thinking about a barrier protecting from infection and water loss, for example. So this is a demonstration that these are generated endogenously, and if you add them back, they can directly, as an agonist, promote tissue reparative processes. Now we know that macrophages are main targets of a lot of these pro-resolving mediators during resolution. And I just very briefly want to note that in response to injury, monocytes are recruited from the bone marrow, and these can supplement tissue-resident macrophage populations in tissues. And they, as I noted, they initially assume an inflammatory phenotype rate. And this is due largely to their exposure of damage-associated molecular patterns and pathogens that are in the tissue, as well as certain cytokines like interferon gamma that activate receptors on these cells, engage transcriptional targets to polarize these cells, if you will, to a inflammatory phenotype. So we're going to be increasing pro-inflammatory cytokines, telling more immune cells to come to the site and things like that. Over time, these cells have quite a bit of phenotypic plasticity. So in response to other cytokines like Th2 cytokines, IL-4 and IL-13, anti-inflammatory cytokines like IL-10 and the resolvins, which I'll talk about a little bit, start to assume different phenotypes that then transcriptionally reprogram these cells to produce mediators that can start to directly promote tissue reparative processes and dampen inflammation or prevent further leukocytes from coming into the tissue. So you can see examples here, a variety of growth factors like IGF-1, VEGF, which is important for new blood vessel growth, and other mediators that can promote epithelial cell growth, for example. So again, these are shown as distinct states, but we understand that in vivo, this is really a phenotypic spectrum, and there's a lot of overlap between these states as inflammation progresses to resolution and tissue repair. So we were interested in understanding how some of these individual pro-resolving mediators engage macrophages to promote tissue reparative processes and resolution of inflammation. So we did a very simple experiment where we take murine bone marrow-derived macrophages and then gave them an agonist. So that in this case, it's a molecule called resolvin D1, and this activates a specific receptor that's called ALX or FPR2. And we carried out RNA-seq analysis and just asked how a naive macrophage can respond to one of these individual molecules. And we sort of classified the genes according to gene ontology analysis and found that indeed this resolvin D1 molecule was able to regulate genes involved in host response and regulation of inflammatory signaling. But we were very surprised to see an overrepresentation of genes involved in tissue vascularization. So you can see here angiogenesis, tube development, regulation of VEGF signaling, for example. So to understand how this pathway may regulate tissue revascularization responses in vivo, we generated mice with myeloid-specific deficiency of this receptor. So this would target largely neutrophils and monocytes and monocyte-derived macrophages. And we used the classic ischemia model that you're probably familiar with, involving femoral artery ligation. This is a permanent, non-reperfused model. And over time, if we monitor tissue perfusion using laser Doppler perfusion imaging, you can see that despite a similar level of ischemia on day 0, that mice with myeloid deficiency of FPR2 they did recover, but they had a significant impairment in the recovery of tissue perfusion by day 10, indicating that activation of this receptor endogenously played some role in the tissue perfusion response. This was associated with an increase in fibrosis that's commonly observed if you have alterations in tissue perfusion. And then we took a cassette of genes that we had identified in our in vitro bone marrow-derived macrophage RNA-seq experiment and assessed their expression in whole skeletal muscle. And you can see many of these are ligands or receptors for well-characterized angiogenic pathways. And many of them were decreased in the whole tissue of the myeloid KO mice. And then we went on to sort macrophages from the muscle to ask whether these genes were altered specifically in muscle macrophages. And you can see a very similar loss of those genes in macrophages. And in contrast, as a gain-of-function experiment, if we take RVD1 and treat the mice, then isolate macrophages, we're able to increase those genes in macrophages, indicating that we were activating those pathways that contributed to tissue perfusion in vivo. Now, since that time, we've been trying to understand the role of these mediators, and particularly lipid mediator class switching from pro-inflammatory to pro-resolving mediators, and how they relate to these distinct events during tissue injury and regeneration. And then so we turned our attention to this model that involves myocyte necrosis induced by a toxin called cardiotoxin. We like this model because, unlike ischemia, it causes a very homogeneous injury and allows us to comprehensively stage the injury and understand the tissue regeneration response, which, unlike skin, in skeletal muscle, it's a complete regeneration and recovery of function rather than a tissue reparative response associated with fibrosis. So I put the cartoon of these dynamic events up here just to orient you again, and I'm going to introduce these markers, Lys6C high and low, which are sort of two mouse markers of inflammatory versus anti-inflammatory or pro-resolving macrophages at the two ends of that spectrum. And we use these to sort of just gauge where we're at in this response. And it's important to note that macrophages are required for tissue regeneration in this model because if you deplete them, you get a very substantial impairment in tissue regeneration, a persistent inflammatory infiltrate, and replacement of functional myocytes with ectopic lipid deposition. So we carried out a comprehensive targeted analysis by mass spectrometry to measure a panel of both pro-inflammatory and pro-resolving lipid mediators. And I'll just point out a few key relationships. You can see that some of the pro-inflammatory mediators generated from arachidonic acid, such as leukotriene B4, peak very early in the response at day 2, and then its levels are basically completely back down to baseline by day 4. Whereas some of the pro-resolving mediators come up in parallel at days 1 and 2, but that they also remain persistent at day 4 when leukotrienes had gone down. So there is some sort of lipid mediator class switching and temporal distinctions that can be observed in this model. And if you just take the ratio of leukotriene B4 to the total amount of these pro-resolving mediators, you can see that this switches between day 2 and 4. So then we asked if we take one of these molecules, like resolvin D2, and add it back to this model, can we impact the temporal dynamics of this response and accelerate the regeneration? So here is a model, this is actually a delayed model of regeneration where we treated with resolvin D2, a synthetic molecule, at day either day 2 or 3 following injury. And then we looked at the expression of Lys6E high versus low as an indicator of those two extreme macrophage phenotypes. And you can see that in control muscle, there's a predominance of Lys6E high cells at this early day 2 time point. And that if you add RVD2, you can shift that from high to low at both day 2 and 3, which would indicate that we're like pushing these cells from an inflammatory to more of a tissue reparative phenotype. But most importantly, this was associated with an increase in muscle function. So this is both twitch and tetanus force that was measured in the muscle following just this 2-day treatment at both day 8 and 14 following RVD2 administration. So since that time, our lab has been interested in understanding the role of this molecule and its specific receptor in regulating macrophage phenotype transitions, and also understanding how the receptor itself is regulated. So RVD2 also binds a specific and unique receptor called GPR18, which was a previous orphan. And if you just look at publicly available data from the Immunological Genome Consortium, you can see that GPR18 is pretty widely expressed on monocyte populations from different compartments, such as bone marrow and the blood or lymph nodes. It's also expressed on neutrophils to a lower degree, as well as a variety of different tissue macrophages, such as those in the lung and in the liver and in the peritoneum and spleen. We looked in our own RNA-seq data set, and so these are neutrophils or macrophages isolated from skeletal muscle during the cardiotoxin injury model at days 1, 2, and 4. And you can see that GPR18 is most highly expressed on Lys6E high, or an inflammatory subset, at both days 2 and 4, and that it increases gradually over time as the tissue repair response is initiating at day 4. So given that it was expressed more predominantly on inflammatory macrophages, but that adding RVD2 switched their phenotype to an anti-inflammatory phenotype, we were interested in understanding more about how this receptor is induced and regulated in macrophages and what its function is. So as I noted, inflammatory cytokines and other pathogen-associated molecular patterns can initiate inflammatory signaling in macrophages that polarizes them to an inflammatory phenotype. So we started by just giving inflammatory stimuli like LPS, which activates toll-like receptors such as TLR4, or inflammatory cytokine interferon gamma. And you can see that each of those were able to induce GPR18 expression in naive macrophages, whereas Th2 cytokine IL-4 was not. And if you add both of these together, which is commonly would occur in an injured tissue, for an example, you can see that there was synergy in that response and quite a substantial induction of GPR18 expression. We expanded this analysis to include a variety of synthetic ligands to targeting different toll-like receptors, and you can see that this is a pretty broad response. Most of these, such as TLR2, for example, 4, 5, 9, are able to induce GPR18 expression, indicating that this pathway may be sort of generally regulated in macrophages as they're exposed to a variety of different pathogens. Now, most of these pathways will couple to a pro-inflammatory transcription factor called NF-kappa B, and a component of which is P65. So in collaboration with Laszlo Nagy at Johns Hopkins University, who's an expert on epigenetics and the role of different gene expression modules in regulating macrophage phenotypes, we probed a ChIP-seq data set that he had on hand where macrophages were stimulated or not with LPS, and we looked at P65 binding within the GPR18 promoter region, and you can see a marked enrichment in P65 binding in cells stimulated exclusively with LPS. And we also noticed a very dramatic reduction in binding of CTCF in macrophages treated with LPS, but not with IL-4, which was sort of consistent with our in vitro data with these agonists and looking at GPR18 expression. Now, just briefly, CTCF it can regulate either transcriptional activation or repression by regulating the interaction between enhancers and promoters. So in this case, it would suggest that potentially this pathway was preventing interaction between enhancers and promoters as a repressor that's then relieved when an inflammatory stimulus is present. So to test causality in the activation of the NF-kappa B pathway and induction of GPR18 in macrophages, we stimulated them with LPS and interferon, as you can see here or here, and just pre-treated these cells with a variety of different inhibitors of the NF-kappa B pathway targeting P65 or its upstream modulators, I-kappa B alpha. And you can see that virtually all of these inhibitors were able to block induction of GPR18 induced by LPS and interferon, indicating that this pathway at least plays a causal role and contributes to induction of GPR18 in inflammatory macrophages. So now to test its downstream function, more specifically in vivo, since we knew that RVD2 treatment could potentially switch these phenotypes, in collaboration with Gabby Fredman at Albany Medical College, she had generated a humanized mouse model expressing a human GPR18 sequence that's flanked by LOX-P sites. And we obtained these mice and crossed them with mice expressing lysozyme M-Cre to remove this gene in myeloid cells, again, just as we had done with FPR2. You can see here that in both resident peritoneal macrophages and bone marrow-derived macrophages from male and female mice, we get a nice reduction in GPR18 expression. This was specific to macrophages, as there was really no significant modulation in splenic T cells, for example. And given that this was a humanized model, it was important to establish that both human and murine GPR18 function in an essentially identical manner, which they do if we compare C57 and our flox control mice in terms of responsiveness to RVD2. They're essentially the same. And we established receptor dependency with Gabby, showing that the effects of RVD2 in promoting phagocytosis, for example, are lost in these mice. So then we turned to two different acute inflammation models with these mice in hand to ask whether this pathway plays an endogenous role in regulating macrophage phenotype transitions to resolve inflammation. And in this model of contact hypersensitivity, where you just challenge with a mediator called oxazolone, which is like a skin irritant. So you sensitize and then you come back and challenge these mice on the ear later, and they develop ear inflammation and swelling that can be readily measured. We carried out LC-MS analysis and found that RVD2 is generated endogenously. So we have the ligand present. So when we're testing endogenous function of the receptor, it could partially at least be attributed to production of the ligand in vivo. And you can see that loss of GPR18 on myeloid cells didn't affect the peak of inflammation, but seemed to affect the resolution phase that was delayed in terms of ear thickness, which is an indicator of edema and inflammation. And when we took the ears, digested them, and did flow cytometry, you can see that there's an endogenous defect in the switch from the Lysix high monocytes and macrophages to the Lysix low macrophages, as shown in these panels here, the highs on the top and the lows on the bottom here. So this indicated that the enhanced inflammation in vivo in these mice is related to an endogenous defect in switching these macrophage phenotypes. To study this in a distinct model to make sure that this is not model dependent, we instilled a TLR2 or dectin-1 agonist called Zymazan, because this pathway has been shown to increase GPR18, as I showed earlier, in a TLR2 dependent manner. And then we just monitored the development of inflammation and resolution in the peritoneum. So this is a very well-characterized model where resolution indices have been established, such as the disappearance of neutrophils from the tissue by 48 hours. So you can see that myeloid deficiency of GPR18 led to an increase or altered resolution response at 24 hours, despite no change in the initial infiltration. And there was also an elevation in Lysix high monocyte infiltration and Lysix positive macrophages, here shown as CD11B intermediate, that accumulate transiently during this response. And then if we take macrophages sorted from these mice at 24 hours and ask how their phenotypes may change, because our previous analysis was limited to just one surface marker, and we know that there's a lot of phenotypic diversity in these different subsets. Again, we collaborated with Laszlo and carried out an RNA-seq analysis and found a cluster of genes that was downregulated and another distinct cluster that was upregulated. And when we looked at gene ontology analysis and to try to sort of categorize the genes that were changed, you can see that one of the most highly changed clusters that was downregulated in the knockout was negative regulation of the immune system, indicating that GPR18 activation increased mediators that would dampen immunity. But we also found some interesting and unexpected pathways, such as lipids and atherosclerosis, and some other interesting categories, such as response to wounding and organic acid transport and extracellular matrix organization, and things that you would typically associate with tissue repair. And so we were interested in looking at these genes in a little more detail. And so this is just a panel of each individual gene that was downregulated in cluster 1. So these would be genes that were lost when GPR18 is deficient. So one of the top regulated genes, just like with FPR2, was KDR, or VEGF receptor, indicating that this may regulate an angiogenic response of these macrophages. And then I sort of just broadly characterize them. So genes in magenta are those that are involved in lipid processing and metabolism, several of which are involved in lysosome function, phagocytosis, and amino acid transport and metabolism. So again, as macrophages are in these tissues and they're taking up dead cells, they have to process all of this cargo. There's a lot of, lipid and protein and things like that that have to be degraded and utilized by the cell. So this may potentially indicate that activation of this pathway regulates that. So you can see, genes that regulate lysosome acidification, and a variety of lipid metabolism genes like lipoprotein lipase, PPAR gamma, LXR, which is known to be activated by cholesterol products, and so forth. In green are genes that are involved in tissue repair and regeneration, such as GDF15 and GDF3, which Laszlo Nagy has shown are important in stimulating muscle regeneration, for example. EGR2, which also regulates macrophage phenotype transitions and could potentially be responsible for some of the observed effects. And then in orange, some of those that are involved in negative regulation of inflammatory signaling, and then there were some genes that were involved in host defense and inflammation, which again is sort of consistent with the role of resolvins in not being completely immunosuppressive. They're sort of immunomodulators that are enhancing certain aspects of immunity, such as host defense, but also stimulating cell clearance and tissue repair. Now, in the last two slides, I just want to mention some new projects that we're working on that are more focused on the vasculature, because several years ago when I initiated some of the first studies that I did with Dr. Sirhan in elucidating the structure and biological actions of RVD2, we had collaborated with Mara Peretti in the UK to do intravital microscopy to understand how this molecule may be regulating leukocyte trafficking during inflammation. And this is data from the cremaster muscle incubated with platelet activating factor. And you can see that co-treatment with RVD2 was able to block the adhesion and immigration of leukocytes into the tissue. And what was interesting is that this was partially reversible by inhibiting nitric oxide synthase. So this is a mediator generated by the endothelium that has very well characterized antithrombotic and anti-adhesive actions. And we found that RVD2 may have some direct actions on the endothelium, but it was something that sort of didn't go, sort of went underexplored for a while as we were focusing more on immunity. And so recently we've been interested in getting back to this and understanding, now that we know the receptor for RVD2, which is GPR18, can we start to understand how it's expressed and what its role in the endothelium may be. So we started and did a survey of GPR18 expression in different human and mouse endothelial cell lines from different vascular beds, as well as lymphatic endothelial cells. And we wanted to know, so we found that it is indeed expressed in all of these different cells, including primary cells. And because we knew that inflammatory cytokines increase GPR18 expression in macrophages, we incubated these cells with inflammatory cytokines and were surprised to find that in these cells, it seemed to be opposite, where GPR18 was more constitutively regulated, but if we stimulated inflammatory signaling, it actually downregulated the receptor. And this was associated with the reciprocal increase in adhesion molecules such as VCAM and ICAM, indicating that this may be sort of being functioning as an endogenous restraint that goes down to allow leukocyte trafficking during inflammation. And interestingly, if we give the ligand RVD2, we can block production of inflammatory cytokines like IL-1 beta, as well as adhesion receptors, VCAM and ICAM, indicating that this pathway could also be harnessed to sort of counter-regulate excessive leukocyte adhesion. And then we wanted to study its endogenous function in more detail. So we started off by knocking down the receptor in endothelial cells using siRNA, which was effective as shown on the top. And we wanted to establish receptor dependency of RVD2's actions, but we were very surprised that as soon as we knocked down this receptor, which again is apparently constitutively expressed, the cells assumed a different morphology almost spontaneously. This is, I think, 72 hours post-transfection. And you can see they sort of lose the cobblestone-like monolayer that typically is observed with endothelial cells, and they're sort of more spindly shaped that are more indicative of a fibroblast-like phenotype. And we assessed genes involved in so-called endothelial to mesenchymal transition that is known to occur in different chronic inflammatory diseases. And you can see that there is an acquisition of some mesenchymal markers such as ACTA2, coupled with a decrease in some endothelial identity genes such as cadherin 5. So this indicated that this pathway may sort of help to maintain quiescence of endothelial cells and keep them in this non-inflammatory quiescent state, and that its loss may drive them towards a mesenchymal phenotype. So this is an interesting new facet of the biology of this receptor that we're currently developing tools to test in vivo. So in summary, what I've told you today is that pro-resolving lipid mediators such as RVD1 and D2 facilitate the transition from inflammation to tissue repair, and this is due in part to regulating macrophage reparative functions. We found that the receptor for RVD2, GPR18, is induced in part by inflammatory stimuli in macrophages, and we've implicated the NF-kappa B pathway in this process. Myeloid-specific deficiency of GPR18 alters endogenous resolution of inflammation and is associated with defects in transcriptional signatures indicative of lysosome function, tissue repair, and counter-regulation of inflammation. And so conceptually, this is sort of another example of the beginning stimulating or signaling the end, right, where these initial inflammatory mediators are sort of priming macrophages to upregulate receptors that are sensors for pro-resolving agonists that then can drive their phenotype to resolution. And then, again, more recently, in addition to its role in leukocytes, this receptor may contribute to resolution in part by regulating endothelial quiescence, which is a new direction that we're pursuing. And overall, the goal of all this is to sort of develop more of a comprehensive understanding of the role of pro-resolving lipid mediators and their receptors, because this may inform novel agonistic therapeutic approaches that are not immunosuppressive, because these pathways actually enhance host defense, and that this may be a better strategy to resolve inflammation and promote tissue repair in diseases associated with chronic non-resolving inflammation. And so with that, I'll end and just acknowledge some of the individuals that contributed to these projects, such as Augustina Mena and Blenda Wong from my lab, funding from the NIH and NHLBI and NIDDK specifically, and then our long-term collaboration with the Nagy Laboratory and Gabby Fredman, who is critical in this work and providing us with some of the new tools to study the receptor. So with that, I'll take questions, and thank you again for letting me share some of our work.
Dr. Speight, thank you. That was just a wonderful, gave us some really intriguing insights into the role of lipid mediators in inflammation and its resolution. And I'm sure there's going to be a lot of questions, but before we start on the questions, let me just remind those who are attending to go to pollev.com and enter DeBakey and respond to that activity. So that's pollev.com if you're on your computer. Enter DeBakey. You can also text us, text at 37607, text DeBakey, and then you can put in your message, and that will get to us, and we'll ask your question of Dr. Speight. I'm going to give, though, the first question to my faculty colleague, Dr. Leli.
So Dr. Speight, thank you so much for your very comprehensive talk. So I guess the question that I'm most curious about is, in your talk, in terms of the animal model, you used mostly acute injury model. So do you think this immune resolving mechanism is different between acute model or a chronic model?
Yeah, that's a great point. So we use a lot of acute self-resolving models so that we can specifically understand the role of these mediators in endogenous resolution, right? So we first sort of have to start in a model that resolves on its own to understand their contribution. And then we can understand, once we know the pathways and have them mapped out, how they may be altered in chronic inflammatory diseases, or whether we can add back some of these mediators to resolve established inflammation. And indeed, now there's been several studies in a whole variety of different chronic inflammatory diseases or models of diseases showing that there is impairments in some of these pathways. And in our lab in particular has collaborated with Gabby Fredman and Ira Tavis's group to show that atherosclerosis, both in human plaques and in mouse models of advanced atherosclerosis, have deficiencies in some of these pathways, and that if you treat mice with advanced atherosclerosis, you can rescue these processes. A lot of that has to do with rescuing defects in macrophage clearance functions. So yeah, there is a lot of evidence now accumulating in chronic inflammatory diseases that this could be an effective strategy.
On that particular point, the role of the resolvins in resolving atherosclerosis, you mentioned that efferocytosis is impaired in the atherosclerotic lesion, and I'm wondering if there's a role for resolvins in rescuing efferocytosis so that debris can be cleared from an atherosclerotic plaque.
Yeah, absolutely. So again, that's sort of like one of the major defining functions of resolution is these agonist-mediated stimulation of macrophage phagocytosis or phagocytosis of apoptotic and necrotic cells in some instances. And we think that's one of the primary mechanisms whereby they stimulate resolution, particularly in atherosclerosis and other diseases, is by rescuing those defective functions. So it's been shown in chronic wounds, for example, and in several different models of atherosclerosis. We've even taken macrophages from diabetic mice that have defects in that process and shown that we could rescue it to some degree. And a lot of elegant work from Ira Tavis has shown that there are certain receptors involved in phagocytosis that are inactivated in athero, and that if you can prevent their inactivation, that can rescue those processes. So it really seems that that's sort of a fundamental mechanism that's at play, and that could be harnessed therapeutically to resolve inflammation for sure.
So I'm also interested in the last part of your work where you find the role of GPR18 endothelial cells. So I'm curious about, firstly, during the like the tissue repairing process, will the GPR18 have the very similar pattern expression pattern similar to resolving?
Yeah, so we're trying to understand that now. So it seems that, well, in immune cells, the receptor is increased by inflammatory stimuli. So in the context of a chronic inflammation, it would be expected to be elevated, which is actually good because then you could target it therapeutically. And what has emerged is that the biosynthesis of a lot of these products seems to be impaired in chronic inflammatory diseases. Not so much that the receptors are deficient. So that's, again, maybe an opportunity to then develop agonists to target these receptors. Now, in terms of the endothelium, we are just now embarking on those studies and trying to understand this, and have just found that it's decreased by inflammation. And so we're developing a new project to start to look at that in vivo and whether there are differences in different cell types. Maybe it's upregulated in the immune cells and downregulated in the endothelium, for example. And there's a whole lot more to understand there, and we're trying to develop tools to interrogate that in vivo now.
So just follow up with this question for the GPR18. So because just if you knock out GPR18 and they're already cause some phenotype in endothelial cells. So, but if this GPR18 is only have a role in immune resolving, but why it has a basal function?
Oh, I see. Yeah, that's a great question, actually. So, again, as I noted, we were surprised, right? So, like, we knocked down the receptor just because we wanted to come back with our agonist and study its signaling. And the cells transformed. So, this was sort of just a discovery that we didn't really anticipate. It could indicate that there's the ligand or other ligands are present in the media or produced by the cells as they're proliferating. There are some of the enzymes that could be present that could produce some of these mediators. But we, again, that's a very recent study, and we're trying to test all of those hypotheses currently to understand whether it's in a, just like an autocrine loop that's automatically or persistently there, or whether there's some other constitutive activity of the receptor or something. But we're trying to understand that now. Great point.
I had the same question. I was so impressed by the effect of knocking out that receptor, GPR18, on the endothelial cell phenotype. Endothelial cells lose their identity when you do that. And they become more adhesive for, presumably for monocytes, because now they're expressing more adhesion molecules. And it does make you wonder if this is there's some basal level of RVD2, these resolvents that maintain endothelial cell phenotype. Fascinating.
I mean, there are other examples, though. It's sort of like the previous concept of inflammation resolving passively versus actively, where agonists are made. The endothelium is also maintained actively in a quiescent state. And there are other pathways, right, like VEGF or FGFR signaling, and the role of the ERK pathway in maintaining endothelial quiescence. And if you just knock it down at baseline, they can, start to change their phenotype. So maybe this is just another one of those examples that there is an active process going on to maintain these cells in that quiescent state.
Another question about, you mentioned that analogs of RVD2, the resolvin, you're starting to work with. Are you planning on developing these into drugs? Are you going to try to commercialize these basic research insights into something that's going to be useful for inflammation and healing?
Yeah, absolutely. So actually, my former mentor, Charlie Sirhan, has really sort of led that effort. So he did the structural elucidation on a lot of these molecules and found that a lot of them are metabolically inactivated in different, a variety of different routes. So they've been trying to develop a lot of different stable analogs to prevent that inactivation and prolong their bioactivity. And indeed, there's a variety of different molecules that have been tested. And there's one of another molecule called lipoxin A4. It's an analog called benzolipoxin that is being tested actually in clinical trials for periodontal disease now, and has shown effectiveness in phase 1 and phase 2 clinical trials. So I think developing longer-lived analogs that could be agonists is definitely a feasible approach.
Because inflammation plays such an important role in healing, of course you need resolution of that, but timing is going to be important in administering these drugs, I would think, because you don't want to interrupt the natural process of inflammatory healing, that role of inflammation.
Yeah, right. So in our acute models, we can control the timing to try to understand how they regulate distinct events. But certainly in the case of a chronic inflammatory situation or a non-resolving inflammation, like a diabetic wound, for example, you would be administering these at a time where those processes are already defective and you're trying to rescue them. So I think that's probably where the therapeutic relevance is. And as I noted, though, like unlike a lot of the anti-inflammatory strategies, and you've alluded to this before too, right? There's a lot of those strategies that can impair tissue reparative responses. Generally that's what happens, and then you can also promote immunosuppression. So that to me is one of the key advantages of these molecules, is they don't seem to promote immunosuppression, and actually they enhance host defense and they enhance tissue repair. So I think that's going to be one of the key therapeutic advantages.
Two quick questions, and I'm going to let you have the last one. Tell us how does diabetes and age affect this process of resolving inflammation?
Yeah, great question. So we've worked a little bit on diabetes and have shown that some of the pathways are perturbed or inhibited in diabetic skin wounds in like leptin receptor deficient mice, for example. Others have shown that different adipose tissues in obese individuals have lower levels of these mediators than their pro-inflammatory counterparts, for example. And we've shown the same thing in other cardiometabolic diseases like atherosclerosis, as well as have others. Aging is a particularly interesting example. There have been several studies now, particularly in mouse models, showing that models of aging do have impairments in some of these products. Even if you challenge an aged mice with an acute inflammatory stimulus, there's an altered resolution. So I think something about the aging process could potentially impair these pathways and decrease sort of resilience to inflammatory triggers that happen in aged individuals. So I think that's also an area that's ripe for discovery and therapeutic innovation.
So I guess this is along with whatever we already asked. It's about because you asked about timing. I'm also more particularly interested in the dosing effect, because I see your previous what the study showed us today. So I don't see the dose a specific effect, but I'm sure you already studied it. But I just want to know, did you see a different phenotype if you increase the dose of the resolving, or because all the knockout mice you cannot get too much information, but for the dose of resolving you should be able to see different phenotype if there is.
Yeah, so certainly in vitro there are clear dose response relationships. And in vivo, the really detailed pharmacokinetics hasn't been established for each individual mediator, because that's quite a large undertaking, and it's something that people are working on and that needs to be done. We've done some limited studies where we've given deuterated analogs of some of these mediators topically in wounds or in our skeletal muscle injury model, for example, and we show that they're very rapidly cleared. So because they're deuterated, we can differentiate them from what's generated endogenously. And in a skin wound, for example, if you give a deuterated analog and you look in the blood an hour or two later, more than like 90% of what we gave is gone already. So they're very rapidly cleared and metabolized. So there haven't really been instances where, we've sort of given too much, I would say. It's usually the struggle is to keep enough there around, and which leads into the point that was made in the past about, developing analogs that are a little bit longer lived. So that's, I think, where it needs to go.
Well, we're at the end of a very enlightening hour with Dr. Matthew Speight, who's Associate Professor in the Department of Anesthesiology at the Brigham Women's Hospital. Thank you, Dr. Speight, for a really intriguing presentation on inflammation and its resolution.
Thank you so much for having me.
Thank you.